Welcome back to the Civic Sci TV News Show. I'm your host, Jake Krauss, bringing you the latest in the world of civic science. This week, we're going on a field trip to the National Zoo, where the Smithsonian is celebrating the summer solstice, the longest day of the year in the Northern Hemisphere, with outreach activities. Zoo scientists and volunteers are showing the public how they use science to protect animals in the wild. Here I am at the Smithsonian Zoo for Solstice Saturdays. We've got Spoonbill here behind me. The Smithsonian Migratory Bird Center gives a demonstration at the aviary on how they band birds with radio transmitters and track them using these antennas and receivers. Visitors handle the equipment to get a better idea of how scientists study the movement of birds, a critical step in their management and conservation. One activity involves matching each zoo animal with its poop, because studying hormones and poop can tell scientists an animal's stress levels or if it's pregnant. One activity shows the hormones as magnetized color beads that can be drawn out from the surrounding blood-colored beads, a simulation of the real detection process. The technician fills the wells with a sample from an animal and performs a test to detect the presence of a hormone, such as progesterone, an indicator that an animal is pregnant. The color change in the sample means that the hormone is present, indicated by the blue color change. The lab technician who does this is the first person to know if an animal is pregnant. Cryogenic preservation of sperm and eggs of zoo animals involves keeping reproductive cells frozen. This can be used for artificial insemination in the future to add more genetic diversity to groups of endangered species if needed, making for a more robust wild population. While it is not safe for members of the public to handle frozen eggs and sperm, scientists demonstrate a minigame where visitors place a bead, instead of an egg, inside a beaker and seal it off. It is actually really challenging using the tongs and is a good test of fine motor skills. Sealing sperm in the long thin tubes is much easier. Props to this scientist explaining her process with patience and for storing the valuable genetic material from each zoo animal. The next step is to keep the eggs and sperm frozen using liquid nitrogen. The demonstration shows how scientists take out sperm from frozen storage, thawing the sample using warm water. This horse sperm, when thawed, can be seen under the microscope and projected onto the big screen. When used for a real insemination, the sperm is filtered and cleaned, but here you can see dozens of sperm cells swimming around chunks of frozen materials and clusters of dead sperm cells. One research project being done by the Smithsonian scientists helping with the demonstration is to find a way to preserve cells without liquid nitrogen, because it is constantly evaporating, as you can see here, and it is quite expensive. This was such a fun day running around the zoo and seeing all the animals and strategies used by Smithsonian scientists to study the animals and help protect their wild counterparts. Thank you so much for watching this week's special solstice edition of the Civic Sci TV Newsroom. See you next time.